king of Indian classical violin, Ambi Subramaniam, with Peter Francis and Pakistan. And now I invite Mr. Chris Gopalakrishnan, co-founder of Infosys and trustee of the Infosys Science Foundation, to introduce our chief guest.
Thank you.
Ladies and gentlemen, it's a distinct honor to introduce our chief guest of the day, a scientist whose work has fundamentally reshaped our understanding of the living cell. Dr. Randy Schekman is a professor in the Department of Molecular and Cell Biology at the University of California, Berkeley, and an investigator at the Harvard Hughes Medical Institute. His journey in high-level bioscience began in Stanford, where he studied the enzymology of DNA replication under Nobel laureate Arthur Kornberg. However, it was during his postdoctoral tenure with S.J. Sanger at UC San Diego that he developed a fascination with cellular membranes that would define his career. Dr. Schekman shares a connection to India through his partner Sabiha Merchant, who's here, who is a professor and Warren C. Evelyn Entor Chair in the Biological Sciences at University of California, Berkeley. She's an elected member of the National Academy of Sciences and was also a juror for the Infosys Prize in Life Sciences in 2017. Dr. Schekman is known for his pioneering investigation in the mechanism of vesicular traffic in the secretory pathway of eukaryotic cells by identifying the genes and mechanisms that govern how proteins are packaged and transported. He unlocked a traffic system essential to all complex life. For his monumental work, he has been recognized with the world's most prestigious honors, including the Geithner International Award, the Albert Lasker Award in Basic Medical Research, and of course, the Nobel Prize in Physiology or Medicine, which he shared with James Rothman and Thomas Sudoff. Beyond the lab, Dr. Schekman has been a titan in scientific publishing, serving as editor-in-chief for both the Proceedings of National Academy of Sciences and eLife. Today, he is applying his expertise to one of the great medical challenges of our time. Since 2018, he has served as the scientific director of Aligning Science Across Parkinson's Disease. This major philanthropic initiative, in partnership with the Michael J. Fox Foundation, seeks to uncover the molecular mechanisms behind the initiation and progression of Parkinson's disease. Specifically, his lab is investigating how the release of proteins into alpha-synuclein may relate to the spread of pathology in the brain. Please joining in welcoming a visionary researcher and the leader in the global scientific community, Dr. Randy Schekman. I request Dr. Schekman to deliver the service.
Well, thank you very much for the generous introduction. It is my privilege to share the stage with our honored young scholars of science and language. I have a connection to Indian science and culture through my life partner, Sabiha Merchant. This evening I'm going to share with you the excitement in two developments relating to the march of molecular biology in our quest to understand genes. I will lead the way to the most recent breakthroughs in genome editing that have been accomplished by many scientists, including my own colleague, Dr. Jennifer Doudna. Although that promises such great opportunities in treating genetic diseases, we are now also faced with an emerging and largely unsolved problem of the march of neurodegenerative disease in the world. And I'll share with you my own efforts to try to help to break the logjam in that problem. Well, let's begin with the great discovery of the structure of DNA that was a milestone in 1953 by these two gentlemen, Watson and Crick, with some vital assistance by Rosalind Franklin, also in London at the time. We realized from their insights and from Franklin's crystallographic structure of DNA that the two strands of DNA can interact by a base pairing mechanism that immediately allowed us to understand how the gene could be perpetuated through life and through evolution. This was a truly monumental discovery. It followed nearly 100 years of discovery prior to that, first in the 19th century with the discovery of DNA itself by a German chemist by the name of Friedrich Miescher, who found this structure in the nucleus of cells, and since it had such a simple chemical composition, for many decades it was believed not to have any role in the transmission of genetic information. It took until the mid-1940s to understand that DNA is the seat of information transfer. It is the blueprint that cells use to convey information from one generation to another. Its structure launched a revolution in molecular biology that has led to a deeper understanding of how this blueprint can be realized, how it can be appreciated by the cells in our body, indeed of all organisms on Earth. These are the human chromosomes that we see here in color codes that are displayed from a cell that is caught in the act of cell division. We can now use the visual inspection of each of these 23 chromosome pairs to detect and to diagnose gross genetic abnormalities. But it took much additional molecular biology work to be able to sequence a gene in order to find the precise point mutations that are the basis of many of the genetic diseases. And now we are at the threshold of discoveries that allow individual mutations to be corrected by the modern approaches of genome editing. Just a primer for those of you who don't remember this from high school biology, but DNA is housed in the nucleus of our cells. It is decoded by an enzyme that makes an RNA copy of one of the two strands of DNA, and that message, that transcript, is conveyed from the nucleus into the cytoplasm of the cell, where it is then recognized by a machine, a ribosome, that stitches amino acids one next to another to make long polypeptide chains. These polypeptides to fold into a unique three-dimensional structure. It's taken scientists using a technique that has been successful in predicting the structure of all protein molecules that ever existed on Earth. Several years ago, they deposited online the predicted structure of 250 million different protein molecules, and scientists now can use this information to do experiments to test the function of proteins. On the other hand, it's also been possible through other investigations to create novel proteins that have never existed in nature, to create them for chemical, pharmaceutical, and technological progress. The discoverers of these programs shared the Nobel Prize in Chemistry last year. Now here is a polypeptide chain. It's a simple one. It starts with an amino acid at one end and is stitched together to make by peptide bonds to make a polypeptide that can grow to great length. Now I'm going to share with you another development that has been long in the making, and that is to understand how cells can react to their environment. In this case, to an arms race that has gone on for over 3 billion years on Earth. You may not appreciate it, but the most abundant particle of life on Earth is the bacterial virus. Every milliliter of seawater has 1 million bacteria, but has 10 million bacterial viruses. Bacteria and their viruses have been in mortal combat over 3 billion years and have evolved mechanisms to evade defense systems. One of those systems you certainly have heard about, and that is a technology that is developed based on the structure of sequences in the genome, CRISPR sequences that can be recognized and can be used to code for small pieces of RNA that interact with an enzyme that chops DNA in a very sequence-specific manner. Now, I want to share with you a video that was done for my colleague Jennifer Dauda on the next slide that describes briefly how this technology works.
Scientists have long
from just 3 or 4 years that has allowed this technology to be harnessed to cure a disease endemic in Africa called sickle cell anemia. Now, many of you know that the oxygen in our blood is carried on a heme-bound protein called hemoglobin. Hemoglobin has 4 protein subunits coded by 2 different genes. It is contained within these biconcave red blood cells. Oxygen is collected by binding to the iron on hemoglobin when blood cells pass through the lungs, in arteries through the lungs, and they convey that oxygen to peripheral tissues where the low oxygen tension causes the oxygen molecule to step off the hemoglobin protein one at a time and to go into tissues to sustain oxidative metabolism. Now, that works very well in most of us, but patients with a rare mutation in one of the globin genes that you can see on the very left, on the top are the first 7 amino acids, the top left, the first 7 amino acids in one of the beta globin proteins, and on the bottom there's a change at amino acid position number 6. This one substitution is enough to cause a critical change in the structure of hemoglobin. This change was discovered by the greatest chemist of the 20th century, Linus Pauling, who predicted the existence of the genetic basis of disease, and he had postdocs in his lab prove that by demonstrating that globin isolated from sickle cell patients had a different electrophoretic pattern than normal globin. Now, what happens in the red blood cells that carry sickle cell globin is that when oxygen is picked up in the lungs and conveyed to peripheral tissues, when the oxygen is released, the globin, rather than remaining a soluble protein, forms a long, rigid fiber, as you see in the lower right. And that can extend through the length of a red blood cell, causing it to rupture and to release hemoglobin into blood. This causes very severe anemia and is a terrible progressive disease in patients who have 2 bad mutations in both copies of the gene. Now, just several years ago, it became possible using CRISPR technology to make a subtle change in the genome of patients with sickle cell anemia to unleash the expression of a gene of globin that is normally only produced in the fetus. And now, with that expression, patients who had sickle cell anemia survive quite nicely because they rely on fetal hemoglobin as opposed to the mutant adult globin. That was the first and most dramatic achievement with the CRISPR technology, but just in the past year, another even more dramatic development has happened that allowed a team of investigators to precisely change a mutation in a young child only 3 months old who would have died within the next few months had that mutation not been corrected to change, to repair a metabolic disorder. So this heroic effort led to the curing of a genetic disease in this one child, and it shows promise of being able to do so for all such genetic disease in the future. This is a tremendous breakthrough. Now, I go from a tremendous breakthrough to something that is rather more problematic and for which there has been no solution and is growing in severity around the world. And that is the basis of neurodegenerative disease. The brain is the great frontier in biology. The brain has tens of billions of cells. Each cell can make hundreds, indeed millions of connections to other cells. That is, we have trillions of different nerve terminals in the brain, informing all processes of thought and emotion. Everything about us is dictated by the selective communication between nerve cells at nerve terminals. Some of these nerve cells die, and as they die, one progressively loses cognitive function. The most severe forms of cognitive loss, cognitive dysfunction in the world are Alzheimer's disease and Parkinson's disease. But it turns out, for reasons that are not well understood, that Parkinson's disease is rising at a greater rate than Alzheimer's disease and threatens to take over many more people around the world. It is a challenge around the world. It shows no, it's like a pandemic. It knows, it obeys no boundaries. It's estimated that in the next decade, China will have over half of the new cases of Parkinson's disease diagnosed in its people, and it's not simply because of better diagnosis, it may be because of environmental toxins. We know from genetic studies that there are 20 different genes, mutations in which cause familial forms of Parkinson's disease, but even those are only a minor fraction of all of the Parkinson's patients around the world. It's likely that 80% of Parkinson patients arise by some sporadic process, which I think is going to turn out to be some environmental toxin. China may be particularly bad in controlling those toxins, but other countries will be severely affected. Now, I'm going to share with you a few observations that have been known now for many years that explain at a kind of a crude level what happens in a Parkinson patient. When a Parkinson patient first presents with the movement disorder, it's estimated that over half of a set of cells in the brain called the dopaminergic neurons that congregate in the midbrain section called the substantia nigra have died. That can be seen visually in the brain sections through the midbrain region in my diagram on the left. The sample labeled C is of the patient who died for other reasons, and this dark band of cells represents the dopaminergic neurons in the substantia nigra that ultimately produce an oxidized form of dopamine that creates the pigment melanin that we know that's responsible for skin color, but this pigment is also present deep in our midbrain and can be seen without staining intersections, as you see here. A patient who died as a result of Parkinson's, as you see on the left, has lost almost all of those dopaminergic neurons, and this proceeds more rapidly than normal, although we are all suffering as we grow older with the loss of these cells, because you'll see in a moment how vulnerable those cells are to almost any stress. On the right is a section of the midbrain that was seen now 100 years ago by a German clinician by the name of Frederick Louis, who found in the dopaminergic neurons in patients with Parkinson's a kind of a clustering of dense material, stained kind of red here in this image, a clustering that is now known as the Lewy body, and it correlates with the progression of the disease. It has only become possible recently to be able to see these in the brain of a living patient with a very powerful and sensitive PET probe. But until now, we could only see these Lewy bodies in sections of deceased patients. Now, one of the proteins that is coded by one of the 20 genes that is known to cause Parkinson's is the gene for alpha-synuclein, and its protein product is shown in a little stick diagram at the bottom. It's a small protein. It's a brain-specific, relatively floppy, unstructured protein. The genetic forms of the disease in this gene are of two types. They are point mutations, such as you see highlighted in red in the beginning part of the protein. Those are in familial forms of Parkinson's. Another is a rarer genetic change that involves the genome acquiring 3 copies of the same gene. Thus, the protein becomes overexpressed. It's more abundant in nerve cells. As a result of either a mutation or of its abundance, this protein tends to aggregate, and its aggregation appears to be the basis of the formation of a Lewy body. So one of the challenges is to try to reduce the expression of this gene or to cause the brain cells to degrade the aggregates. That is a major challenge that we face. Now, more recently, another German clinician, Dr. Brock, sectioning many brains of patients who died at different stages in Parkinson's disease, observed a progression beginning on the left, as you see, with a few Lewy bodies that can be seen at the brain stem. Lewy bodies in the olfactory bulb. Now this is and not just in the dopaminergic neurons, dopaminergic neurons of the substantia nigra. Now, the alpha-synuclein is just one part of a very complex story. There are many other genes that are involved in this disease, genes that operate in many of the other cells in our body. So why do these genes, when mutated, affect dopaminergic neurons so selectively? Well, one piece of evidence for this is the environment between dopaminergic neurons and cells in the striatum that perform all of the functions, executive function, movement functions that we know, are afflicted, are compromised in patients with Parkinson's. These cells are poised on the edge of death. They must survive. They must sustain themselves by energy production, and any little insult is likely to propel them over the boundary